Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Christin Burd. We are going to have a really great show for you all. We're going to be discussing epigenetics and how it relates to cancer and different outcomes of cancer therapies. Of course, we will relate it back to those biological age outcomes and other insights we may learn about our health. We'll also be talking about a really great new senescent cell predictor and how that was created using epigenetic methylation markers. An introduction into Dr. Burd. She grew up in Cleveland Heights, Ohio, and received her PhD from the University of Cincinnati, where she worked with Dr. Karen Knudsen, now the CEO of the American Cancer Society, on prostate cancer. Following her postdoctoral training with Dr. Ned Sharpless, the former NCI director at the University of North Carolina at Chapel Hill, Dr. Burd joined the Ohio State faculty. She is now an associate professor in the departments of cancer biology and genetics and molecular genetics at the Ohio State University James Comprehensive Cancer Center. Holding appointments in both the undergraduate college and medical school, Dr. Burd's research is very diverse. It ranges all the way from cell biology to biochemistry to even animal models and human clinical trials. Her main research focuses on identifying mechanisms to prevent melanoma and improve clinical outcomes in older adults with cancer. Her contributions to science include discovery of the very first human circular RNA associated with disease, even the development of P16LUC mouse model to track aging, and spontaneous tumor formation in live animals, and even the establishment of the mutant-specific role of oncogenic NRAS in melanoma formation. In recognition of her work, Dr. Burd has received both the Damon Runyon Innovation and Glenn Awards and currently serves on the scientific advisory councils for the American Federation for Aging Research. She's also serving on the Pan-American Society for Pigment Cell Research and the NCI RAS initiative. Her ongoing projects in the Burd Lab aim to prevent NRAS mutant melanoma and understand the impact of T-cell senescence on health and longevity. Dr. Burd is going to offer a very unique view on her research regarding her very diverse background. And we're also going to talk about advice that she would give to people navigating the science world or who are new to it and may not have any resources to go to. Dr. Burd is going to be offering you all a wealth of knowledge on epigenetics and cancer. I hope you all enjoy. And now for my guest, Dr. Christin Burd. Welcome to the Everything Epigenetics podcast, Dr. Burd. I'm excited to have you here today.
Yeah, this is great. I'm so happy to be here with you and to talk a little bit about our research together.
Yeah, absolutely. So I don't know if I've ever told you this before, but I'm actually from north of Dayton, Ohio. So grew up as a Buckeye, really small town called Piqua, but, loved watching Ohio State growing up and all of their sports. So when I remember first talking with you or sending emails back and forth with, True Diagnostic, I was like, this is a dream come true. I can't believe we'll be working with, someone with your expertise from OSU. So before we hop into more of the research and our larger discussion today, can you just give our listeners some background information about yourself and, especially your expertise in cancer, what led you to become interested in this and the career you have today?
Yeah, sure. So, as you mentioned, I grew up in Ohio. I grew up in Cleveland Heights and went to Cleveland Heights High School, which is now famous for the two Kelsey brothers. That was the same high school I went to for the Kelsey Super Bowl face-off. They're a little bit younger than I am, but not too much, we'll say. And that's where I really got interested in science. And I think a lot of students get interested in science in high school, but they don't know what to do with it next. And I was one of those people. I went to college thinking I want to use science to better the world. And really had no idea how to do that. I think I tried out, a lot of different majors trying to figure out where my fit was in those different majors, thinking, you have to be a physician or a physical therapist or something like that to make an impact on people's lives. But when I got into the laboratory, I just fell in love. It was an eye-opening, an amazing situation where people were interacting and talking about the science and trying to figure out really complex problems and how to solve those. And no problem is more complex, I think, than cancer. I mean, this is a cell of our own body that has basically changed itself to become kind of an evil nemesis. And how do we distinguish? How do we teach our bodies to find those evil cells among our healthy, normal cells? Very challenging how to combat this. And so that was the big thing that drew me to cancer research, was just the complexity of the problem, the number of people that are affected, and just the wonderful cancer research community that's out there.
Yeah, that's great. That's such a good way to explain it as well. The cells of our own body really, betraying us, so to speak, I think is a good way to put it. But you couldn't be more right regarding the science major comment, right? I was the same way. I went, animal science, then biology. I was like, wait, that's probably too general. Do I do biochemistry, chemistry? I mean, physics, who knows? So we'll talk about that at the end there, and maybe, what will help navigate some people going through the same thing, because that can be difficult. But moving more toward our conversation in epigenetics and cancer, in 2020, you actually published a paper on epigenetics as it relates to age and cancer. So I'd love to hear a little more just, what you're doing now and the main mission of your lab as it currently stands, to give our listeners a little bit more insight. True Diagnostic and Dr. Burd and all of the work that you've been doing. We've been doing some things for quite some time, and we'll talk about those projects here soon too.
Yeah, so I have for a long time been interested in the connection between cancer and aging aging is a risk factor for cancer. When you get older, you're more likely to develop cancer. But cancer therapies also accelerate aging. So it's kind of this circular issue where you get older and you're more at risk for cancer than you get treated for cancer, and that makes you even older, and that causes other problems for health span. And so we've been really interested in understanding both that initial part, what about aging makes you more at risk for cancer, but then also what therapies are causing aging, how that process might be mitigated. And, it's really wonderful to see so many cancer therapies coming out and patients have options. And I think now we really need to think about, during the course of that selection of options, what is the long-term effect of each one of those therapies for people? Not just, here's 4 different potentially curative options, but which option is going to give you the best healthy outcome long-term? And so that's kind of where we started to partner together with True Diagnostic, because you have such a wonderful cutting-edge biologic clocks that we can use, the epigenetic profiles, and, great bioinformatics support, meaning that we can take some of the knowledge about the changes in the epigenome and then dig through it to try to understand what the underlying processes are that are causing that change. And I think that's what's really amazing with partnering with you, is that we can get at the biology, what is causing these changes, not just measuring them. So that's been a fantastic collaboration.
Yeah, I know you've been in touch with Varun Dwarka. He's our head of bioinformatics at True Diagnostic, and then Ryan Smith as well. So some colleagues, and we'll continue to learn insights as these interpretations grow and we're able to uncover more. So super excited to be working with you on that. I want to talk a little bit more broad picture, and then we'll go really into the specifics, because there's so much exciting work that you're doing. But we essentially are using the epigenetic methylation markers and, interpreting them for age outcomes and some other insights as well. But why do we even need these biomarkers in the first place for adults with cancer? I know maybe you can connect that then to the therapy outcome or different, cancer therapies.
Yeah, so I'll give you the perspective from our clinical team. So here at Ohio State, we have a clinical team that works in a specialized clinic called the Cancer and Aging Resiliency Clinic, so CARE. And what the CARE clinic is a multidisciplinary clinic, meaning that there are lots of different types of practitioners that visit the clinic. And so a patient will come in and we will holistically look at their health. So we'll look at the kinds of medications they're taking to make sure there aren't interactions. We'll look at their cognition. We'll look at their physical function and fitness. We'll look at their social support networks and try to build those and increase those. But at the same time, we're going to look at these biomarkers and of aging. And one of the reasons we want to do that is that we recognize that patients are often excluded from clinical trials or excluded from particular therapies based on their chronologic age. So just, what your age is on your birth certificate, based on your birth certificate. But we all also know people who are 80 and running marathons, right? So these people are probably more fit than I am. And so we really need to think about how we're making those cutoffs so that we can ensure that we're giving the best therapy to everyone, but also that in the individuals who may be more at risk or more frail, that we're not causing long-term disability by a particular treatment option. So this is really where these biomarkers are critical in decision-making. We can't just assume that a clinician can look at a patient and decide, oh, this patient's going to do really well on this therapy and, they'll come out great. There was a study done in older adults over 60, and they asked older adults, if we had a cancer, all these older adults had cancer, and they asked them, if we had a cancer cure for you that would cause disability, would you take it? And 70% of them said no. They did not want to live with a long-term disability. And so thinking about that, right, is it worth it to cure your cancer if you can't live on your own? And I think for older adults, quality of life really matters. And that's why these aging biomarkers are really going to help us improve the quality of life for our patients.
Yeah, that's interesting. So you're using these epigenetic markers as a way almost for like inclusion and exclusion criteria for different studies with the people who have cancer, of course. Is that correct?
Yeah, so that's how we want to do it. I mean, right now we're working to pair that and look at outcomes to figure out, will it be more informative for physicians? For the most part, what we're finding is that a lot of people are being excluded that shouldn't be. And so I think it will lead to more inclusiveness, which is great. But also we do want to prevent, that small number of people who we wouldn't predict would have a really terrible reaction to their therapy. If we could tell them, yeah, we can still give this to you, but, maybe it would be better to use this other therapeutic option that's known to have fewer side effects. So those types of decisions have to be made on the fly every day in the clinic. And they're often just clinician specific, they call it the eyeball test, right? They're just looking at the person and thinking, oh, how frail or fit is this person? We need biomarkers. We need quantitative measures. We don't want people just giving us a once over and making decisions.
We need to remove the guesswork, right? The eyeball test, that makes me cringe almost.
It makes them cringe too.
Right. They know it. So I think it's just, incorporating something better, right? Kind of just to make those studies even better and have better insights, right? To make changes in different assumptions. So no, that's great to hear that you all are working on that for that reason. Changing subjects or going a little bit deeper, I could say. A lot of the work you're now doing is with T cells, right? So that's a big part of what True Diagnostic and your team have been collaborating on. So What about T cells? Can we use these types of cells to actually measure epigenetic age? And, we may have some listeners who aren't super familiar with T cells as well. So if you want to give an introduction to T cells, that would be great.
Yeah. So in our blood, so most of the epigenetic clocks that have been developed, or the measures that we use, have been based on blood or tissue samples. And in your blood, there are all kinds of different cells that are circulating. Some of them are your immune cells. And even among those immune cells, there's all different types of immune cells. There's immune cells that attack cancer cells. There's immune cells that suppress autoimmune diseases. There's all different kinds of immune cells that circulate. And when you get an infection, you probably realize this, as those cells will expand and build their armies, right? And then you'll have swollen lymph nodes, or maybe even a fever from them getting activated. And one of the key immune cells that's involved in both responding to infections as well as responding to cancer and killing cancer cells are these cells called T cells. And so we're really interested in T cells as they pertain to cancer and cancer therapies, especially immunotherapies. A lot of the immunotherapies that we're using now, these new generation molecules, they target and try to activate T cells to kill your cancer cells. And so we thought, well, if you're measuring epigenetics in all the blood cells, and we know that your blood changes as far as what different types of immune cells are in there on any given day, right? We may just be measuring changes in the populations of cells that are there. Mm-hmm. But if we can focus in on a specific cell type and understand how the epigenetics of that cell type are changing over time, or the aging of that cell type is changing over time. And we picked T cells because of their importance in cancer and immunity. And so that's where we began to do this research. But can you measure epigenetic age in T cells? Getting back to your question. Well, we had to really partner with True Diagnostic to figure out the answers to this question. And what we did was we were able to take blood samples and then purify T cells from the same person and send them to True Diagnostic. And they did the analysis, and then we compared the results. And indeed, you can measure aging in T cells. It gives you a little bit of a different number than if you just measure the blood. So it's telling us that there is something different between T cells and the blood. But we are working together and partnering with True Diagnostic to really understand what this means for patients and why these differences occur, so that we can, better tailor our therapies and outcomes.
Yeah, it's so cutting edge. It makes me smile from ear to ear. I think looking at the specific, epigenetic methylation markers and outcomes in the different cell types is the future of epigenetics, right? It's already been discovered that there are differences. So now we need to understand what those differences actually mean, right? So I think, again, I just love the work you're doing and how we can start to relate this to different clinical decisions or outcomes. So I know, you've even looked at these T cell epigenetic ages and its relationship to maybe things like clinical measures of, fertility, cognitive decline, and toxicity risk. So are there any correlations there?
Yeah, so we're still building out those data sets, and we are starting to see some signals where, in small cohorts, there are definitely associations between the epigenetic clocks that we're measuring and things like frailty or risk of hospital readmission, things like that, neurocognition. So as we build these out, we're going to have to expand them to additional centers around the United States so that we can capture larger populations, and we're starting to do that now, which is fantastic. We're expanding, we're starting some clinical trials to really understand if we can validate these such that they can be used on a regular basis in clinics all over.
Yeah, and what cohort are you using for that, or what patients? Can you talk a little bit more about the patients that you're looking at?
Yeah, so our biggest focus right now has been on myeloma patients because of we know that there's a lot of accelerated aging that goes on with the treatment for myeloma, which is often an autologous bone marrow transplant. So they're taking their own immune stem cells and removing them from the body, irradiating the person so that that person's cancer cells are killed, and giving them back their stem cells to repopulate and remake their immune system. And that process is really dramatic and causes a lot of accelerated aging. And so we've been really focused on that particular group because we know it's such a problem there. And then the next group that we've been looking at are individuals with what we call solid tumors. So those that don't float around in your blood, and those would be things like lung cancer and melanoma, where we are using immunotherapies because obviously we're really interested in T cells, and T cells are what's being targeted by a lot of these immunotherapies. So those are two major cohorts right now that we're looking at. And all of our patients enrolled in these studies right now are 60 or over, because we are trying to capture some of the functional deficits that occur with age.
Sure. And I forget the name of the first cancer therapy you mentioned, but all of these therapies you're using, that was my next question. Do they show a link to epigenetic age? Or I don't know if you can, maybe
Yes.
go over each of those therapies.
Yeah, so really interesting. For each cancer type and for each person, the therapies have become more and more individualized, right? So there are a lot of different decision-making points along how we decide who gets what. One of those is the genetics of the tumor. So what types of genetic mutations are present? Another is a person's chronologic age, which we'd like to, eliminate. And then the type of cancer, obviously. So we have people receiving all types of therapies. What we have found is that for individuals receiving chemotherapy, if you measure their epigenetic age before they start chemotherapy, and then even long after, like years after they receive their last dose of chemotherapy, epigenetic aging is very prevalent, and you can see that in their blood. And we also see that with immunotherapy, which is pretty new observation. I don't think a lot of people have looked at this yet, but that may mean that people who have been cured of their cancer with immunotherapy may be at risk for other types of age-related diseases. The one thing that's super interesting, though, is that we've seen some patients are treated with a demethylation inhibitor. So their cancer type is actually treated with a drug that inhibits DNA methylation. And we see that those individuals appear to be doing better after therapy. So we're wondering, is it improving their epigenetic age? And if so, how? And could that be applied to healthy people too? If this drug were implemented for short term in a healthy individual, would it potentially reverse aging? And we'll probably talk about that more later and our goals in reversing aging. But yeah, that's one really cool finding we've had, is that there are some, very specific targeted drugs that seem to have kind of an anti-aging effect.
Gotcha. Interesting. So you said that drug, hold on to that point for a second. You said that drug is going to remove methylation?
Yes.
So give you the expression of those genes and keeping those genes turned on, essentially. Mm-hmm. Yeah, that'd be interesting. Have you done anything in a healthy cohort then with people without cancer in that drug?
We have not. So this is really the first piece of first observation of this in a cohort. Not many longitudinal studies have been done, meaning that you take the same person and you follow them over time as they start therapy and continue therapy. So we have one of the, our clinic is really nice in that we can have patients come back time and time again and really follow what's happening with them. And that's what's led to this kind of unique discovery. And, as we try to understand the mechanism by which that is happening in collaboration with True Diagnostic, we really hope to bring that forward to apply it to more people.
Yeah, that's exciting. I think a lot of times when you're treating these, I don't know the right word to use, maybe more disease-based populations or cohorts, and you're trying different therapies, seeing what works, you see some really great effects, like they're doing now with the lithium in people with bipolar disorder. They actually, think maybe low-dose lithium could have some reversal with these epigenetic age clocks, even though lithium has been studied for a very, very long time in aging. So it's great that you have this cohort, especially with the care you all do. You're able to get them back in the clinic and do some, longitudinal testing, like you mentioned.
Yeah, and then pair that, right, with how are they feeling? What is their function like? So I think it's really important that we show that these epigenetic markers are improving the physical function and lives of these people as well. So that's what is nice about longitudinal studies.
Yeah, of course. Yeah. And I can't believe, I know I didn't comment on it, but yeah, you said the 70% of people who took that survey said they would, not want a therapy if they were to be disabled later on, right? To get rid of that cancer. So I think that's huge too, right? They want something that works, and, they want to be cured. They don't want to have any side effects. So really paying attention to what the patient wants, right, is definitely key. Mm-hmm.
Yes.
So how did the patient epigenetic age relate to different outcomes? I know you may have hinted on this a little bit, but can you expand that?
Yeah, so we have not seen any dramatic change that's associated with the outcomes yet. Of course, all of our studies are somewhat limited in how long we've been following people. A lot of cancer studies really require 5 years, sometimes even 10 years to figure out, how many of those people had disease recurrence, how many of those people remained healthy, did they succumb to other diseases? Also, COVID has not helped things, and interpreting data, it's made it much more complex. So I think that, we'll know in the future, but right now, really early on, we haven't seen a specific signal. But, again, it really takes a long time for those data to mature.
Sure. We'll learn. And yeah, of course, COVID-19 adding another variable or layer to look at there. So it doesn't make it easy on you all, or, the people who are actually doing a lot of that data mining. We're going to switch though to senescence. So I think probably almost everyone listening is familiar with senescence, right? It's this buzzword. It's really popular in aging right now. And it's one that is notoriously difficult, as what defines a senescent cell. This is differentiated by cell types. So we're, I think, still learning a lot about this, even the definition of this word. Can you explain senescence? And then can you talk about how these senescent markers are involved in your work?
Yeah, so senescence was discovered a really long time ago when researchers started growing human cells in the laboratory. And what they discovered is that after, so many months of growing them in the lab, they would just suddenly stop growing. And they were trying to figure out why this was happening. And what they found out was that at the end of our chromosomes, there's some repetitive DNA sequences. They're kind of like insulators that sit at the end of each chromosome. And every time a cell replicates, they get a little shorter. And if they get too short, then the cells actually start to lose their genetic material because the insulators on the end fall off. And so these telomere shortening processes would lead to these cells not growing anymore. And they would just sit there in the dish. They would sit there for months on end doing nothing. And so we, started to notice that this occurs with lots of different cell types. And then people began to find them in the body. That, as we aged, you could see that some of these cells were appearing in almost every tissue. And that led to this idea that these were probably negative in some way, these senescent cells. So senescent cells kind of can occur in response to various stressors. So for instance, if you have a gene mutation that could lead to cancer in a cell, sometimes that cell will just become senescent, which is great because you don't want a cancer. It basically will sit there, it won't divide, it'll just kind of chill. I like to draw senescent cells as like sleeping on a pillow. Like,
Oh, there you go.
they're dormant, they're kind of just hanging out, chilling. But the problem is that they don't help either. They're kind of lazy, right? So if you have tissue damage or injury, then what happens is they don't help to repair that. And so that's a problem. We have all these kind of bystanders just standing by looking at the damage and not doing anything. We've also learned that senescent cells can make various signals that increase inflammation. And inflammation is not a great thing. When we don't need inflammation in our body, we don't want that going on. So what we've done is try to understand the molecular changes that occur with senescence, but those are very different in a lot of different cell types. So that's why there's so much confusion about what is a senescent cell, because I think if you look at any given cell type, how it adapts and how it decides that it's going to, become this sleepy bystander is a little bit different. There are lots of different metrics out there that we are trying to apply. Things like the expression of a tumor suppressor gene called P16, sometimes used. Shortening of those insulators on the end of the DNA called telomeres can be used. But these often need to be paired with other aging biomarkers like an epigenetic clock or things that will measure some of these inflammatory cytokines.
Yeah, so just so we can learn more about them, you mean?
Yeah, it's just because they're so complex. Not every senescent cell is exactly the same thing, right? So we can't pick one particular biomarker and measure them all, if that makes sense.
Yeah, definitely. And these senescent cells, I always hear them referred to as these zombie cells, right? I think it's so, such a buzzword I mentioned just because people are realizing like, oh, we have these cells that you said are dormant or are sleeping, what do we do with them type of thing, people who are really involved in their own health or maybe, quote unquote biohackers. So you said the senescent cells, I really liked how you described them as bystanders. Are any senescent cells good, or do you want, none at all, or do we not know? Do we need more research?
Yes, so senescent cells actually play some important roles in things like wound healing and things like, processes like that. So they are important. And they're also important if we didn't have senescence, you would be at high risk for cancer because our genomes aren't perfect. And even in replicating cells, we make mistakes or just going outside, environmental carcinogens, whatever we're exposed to in life can cause genetic mutations that would promote cancer. And most of the time, what our body does is it responds by triggering senescence. And so, that's why we don't constantly get cancers, right? So senescence is good in some ways, but I think this long-term accumulation over a lifetime, once you start getting up higher and higher and the frequency of those bystanders gets higher, then your ability to recover from things starts to suffer. And that's the problem.
Sure. We maybe need a healthy balance, whatever, healthy means, right? We don't want too much for the inflammation, indication of cancers, but, we don't need too few either. We need some standing by to again help remove that cancer. So interesting, and I know a lot of researchers out there are doing work on different senolytic therapies and maybe how to clear out some of those senescent cells as well. But I want to talk about, you mentioned the P16. So that's a really exciting part of the research we're doing with you. So maybe I know you mentioned their tumor suppressor protein. They play a really great role in cell cycle regulation. But do you just want to define P16, and then sometimes it's also P16 Inc4A, correct?
Yes. So P16 Inc4A is its full name. The reason that it's called P16 is based on the size of the protein. So there are other proteins that are the same size. So if you go into another field and you're not in a senescence field and you say P16, they may mistake it for something else. So that's why sometimes you see INC4A put on the end, just to kind of key in, right, what we're talking about. What P16 does is it's turned on, it's a gene that's turned on when cell stressors are detected. So we have almost none of it in most of our cells. But if we have some sort of cell stressor that comes along and triggers that, like a gene mutation that would lead to cancer, P16 gets turned on. And what it does is it halts the cell, keeps it from dividing until it can either fix the problem, or if it can't fix the problem, after a while, it will essentially put it to sleep. So it becomes a bystander cell and becomes senescent. So what's been interesting about P16 is that when we realize that P16 increases with age, people said, oh, and it increases in senescent cells. We thought, oh, this could be a surrogate of how many senescent cells you have in your body. And now lots of studies have been done linking your T cell, how much P16 your T cells make, to things like your fitness or frailty, your risk of hospital readmission, and things like that. So we're beginning to understand P16 as a surrogate for how many senescent cells are in your body.
Gotcha. And maybe backing up, is there a current way we can actually measure the number of senescent cells in your body?
Not perfectly. I think we can get
right.
back to, right? That question that you asked about how do you measure a senescent cell, and there's no perfect way to do that right now.
Perfect.
So, yeah, and in the past we've really focused on T cells because they were easily accessible, from a blood draw, and that's where a lot of the P16 data comes from.
Yeah, and that was, I wanted to ask that question before I asked this one, because again, why your work is so groundbreaking. So are these the P16 levels in these T cells, are those equivalent with epigenetic age measurements or those senescent cell measurements? Is that a way maybe we could start to learn more about that?
So very interestingly, in partnership with True Diagnostics, what we've been able to do is take T cells from the same individual and look at the P16 levels and then all the different epigenetic clocks that you're developing in other groups. And they appear to be different measures. So they don't necessarily match up. But why is that exciting to us? Well, that's exciting because we think that they're measuring two different types of aging, right? They're measuring two different occurrences. And if we could build an epigenetic clock that would predict P16 levels, then we could kind of integrate those two types of aging into one metric. And that's what we're trying to do. It sounds very complex, but essentially, as I described before, aging and senescence can be, measured in many different ways. And if we can capture as much of that complexity and put it into one metric, it'll make it much simpler to kind of judge outcomes for patients.
Yeah, and I think that initial data is very, very exciting. So I think we're, hopefully getting a lot closer to, creating one of those predictors, essentially, which would be huge, right? We would be able to then, you'll be able to talk better about the application than I could, but be able to look at, maybe those different cancer therapies, look at different just interventional therapies for, more of your healthy populations and see maybe what's reducing senescent cells or, what may be working in a way that we thought it wasn't before. So I think that is again, something that is super exciting.
Yeah, no, it's great. I mean, one of the things that's been really challenging about P16 is it's hard to measure. We are using RNA to measure P16. It's not as stable as DNA out in, normal conditions. In the laboratory, we can, stabilize both. But so if we want to reach people who are in areas where they're using a community hospital, how are we going to measure P16? Very challenging. And I think by using DNA and epigenetic-based measures of P16 or surrogates of P16, we will be able to reach people all over the world. And that's something that we would love to be able to do, and to really measure senescence in a more global population, not just in the people who live close to a laboratory.
Yeah. I think that's a great point as well. We want to make sure that everyone can use this type of testing, right? Epigenetic methylation in general, that the testing is so new. It's definitely not the cheapest. I understand that, firsthand, but hopefully just like genomics, now you can sequence your entire genome for less than $100, less than 24 hours. And hopefully epigenetics will get there one day, but the insights you can learn from, looking at an entire array, about 1 million of those methylation markers is phenomenal. There's so many different insights. So very cutting edge, very exciting. And I'll wrap up more of the research talk, and then we'll talk more about, you as an educator at Ohio State. But what's next for you in the research realm? Are you, trying to wrap up some of these studies, or, what are you working on right now?
So I'd say in terms of these studies, we are trying to build that senescence clock and really test it, right, in multiple populations. So that's one angle. The other is expanding some of these observations that we've seen with epigenetic clocks and outcomes in patients to include other universities and other sites, so that we can really understand the applicability broadly. And then I'd say the third area is, how do we reverse this aging, right? And one of the keys is you can go back into this epigenetic data, as I alluded to before, and start to pick out the genes where this methylation mark is modified. And we can go back in the lab and say, well, is the change in the expression or the methylation of that gene causing aging? And so then how would we turn that off or turn it back on? So these are the types of questions that, will be long-term in my laboratory to really, we want to have a long health span in everyone, not just cancer patients, but really to promote healthy aging for everyone.
Yeah, definitely. And I'm equally excited about all three of those points. I don't think many people realize the importance of the third point you mentioned, which is basically we're looking at the differentiated methylated regions. So on all of these methylation, patterns we're seeing, which genes are they affecting? And if we can understand that, then maybe we know the mechanism of action that, whatever therapy or kind of insights we're looking at is working by. So I just think that's so valuable. I think that's just so exciting as, this epigenetic field grows. So we'll see, but I love all of the work you're doing, Dr. Burd. It's amazing. Shifting more to you, I'm excited to learn a little bit more about what you do, though, from an educational standpoint. I know, previously before we talked, when we were doing this recording, you mentioned to me that the diversity and inclusion, all these initiatives, the DEI are super important to you in science. So, what are some paths forward for those who have an interest in this field, but they don't really know how to get involved? Maybe students or people of all ages, I guess.
Yeah, for sure. So, I think this is an important topic to me because I see that there are students that enter Ohio State or people that I interact with in the community that have amazing interests in science, but don't have someone in their family or don't have, a connection to a scientist. And so navigating and getting those same opportunities is incredibly important. So how do you, if you like science, like I did as a high schooler, but you have no idea what you want to do, like we both did, then I think the best thing to look at are some of the programs that are offered through the National Science Foundation, through local colleges and universities, including community colleges. And you don't have to live near them. There are scholarships that are available to pay for your housing, to pay for your food, to get everything so that you can try out science. And you don't have to have experience in the lab to apply for these things. The thing that they're looking for on these applications is interest. Okay, so don't count yourself out. If you're interested in science and you don't have the skills yet, that's all the reason to apply for these programs. And so I am going to give Hannah a whole bunch of listings to put with this podcast so that you can go online and look. These programs start at the high school level. There are programs also at the collegiate level. And then there are programs after college. So if you decide maybe research was something that I wanted to explore, but I just didn't have a chance or an opportunity during college, let's do that, get that out there. And then finally, for those of you who love science and are not necessarily college age, I'd like to encourage you to take advantage of a lot of programs are out there at universities. Like at Ohio State, we have a program 60. So program 60 is for anyone over 60 can enroll for free credits. And you just come and audit the class. I've had several people come and audit. They've been some of my most exciting and interesting students. They have just such an amazing life story, and they'll bring in their knowledge and background and really add a lot to the course. So look at your community colleges and public and state colleges for those opportunities. Some of them are even online, so you can access them anywhere. Just
that's great.
And find them.
That's great. And yeah, just get started, right? I remember I did some undergraduate research at the University of Kentucky, but it was so hard, right? Because some, when you start to get into your major and you're trying to navigate it, I was a sophomore, I was a junior. I was like, oh, it may be too late because a lot of labs want you on for, at least a year or two, right? So just start looking at, those resources available. I'll put everything in the show notes that Dr. Burd mentioned. Funny enough, I just went to a career fair to try and hire some students and get some interns this summer at UK earlier this week. And, what's I think even more disappointing is they grew up through COVID, right? They may not have some laboratory experience, right? So that's something I thought about during when we were there, and I was like, oh man, they haven't gotten the opportunities that people have in the past. So important to look at places like the NSF or local colleges. I think that's great advice. I even remember applying to colleges and being so nervous and scared. I applied to Ohio State and Kentucky, decided to get out of Ohio and, drive 2.5 hours down I-75. But I was torn between the two. I really was. I currently have little cousins going through this process, and, again, my parents didn't have a scientific background at all. So I had to navigate the field on my own. So maybe for any younger, students out there, or even parents of students, what's just one piece of advice you would give to them, if they're going through this process, or even the application process for college right now?
Yeah, I think every college kind of looks for something different, and it's hard to know what exactly to write. But I think in the end, if you're true to yourself and you write where your passions lie, that's going to help you with, finding a match. And maybe you think the match is Ohio State, but if you don't get in, maybe that really wasn't the best place, for your growth. The other thing I would say is, make connections with people. Don't be afraid to reach out to individuals at universities that you're interested in. I'll frequently get emails from people saying, hey, Dr. Burd, can you tell me a little bit more about the Department of Molecular Genetics where I work? What kinds of courses do people take? What kinds of things do people, after they get into molecular genetics, do with their lives afterwards? What do your students do next?
Yeah.
So
That's awesome.
yeah, those are questions that you can reach out. We're just normal, everyday people. I think that's one thing. People are like, oh, they're the professors, but we're really everyday people, and you can contact us and reach out. All of our, if you look on any of the web pages, there'll be contact information to reach out and get more information. So don't be afraid to do that. And, putting your name forward can be really important.
That's great advice. I know I was so nervous. I was like, I'm just a young student. They don't want to hear from me. And that's just, not true at all. I even love when people reach out to me, from True Diagnostic or Everything Epigenetics. Hey, what do you do? What do you spend your time doing? And it's enlightening to talk to my cousins who are going through this process and ask me those questions. Do you sit on the computer all day? What do you do? And no, I think that's great advice. And really, one of my last questions here, Dr. Burd. I've briefly chatted with a lot of other people on my podcast about, their research projects and how they're funded. But I know you have a lot of experience in this realm. So, you mentioned to me that this is an important point, because I think the general public doesn't understand how hard it may be to get funding for these types of projects that you're actually doing. I remember when I was an undergrad and I would go to my professor's office for their office hours, they would have like a piece of paper that says, office hours canceled, working on grant. And I was like, come on, it couldn't be that hard, right? We have a full-time grant writer here at True Diagnostic. We all review the grants. We spend a lot of time making sure there are no mistakes. So I definitely understand why they had to cancel their office hours. So tell me about the process, like what, and why do you believe there's like this lack of general understanding? I know I definitely don't understand it to the fullest either.
Yeah. So I think some of it, scientists don't talk about it. Right? We don't talk about everything. But we don't go out into the general public and say, let me tell you about how I get funding. And so it's kind of a mystery, but in general, funding is either run through foundations. So you may have heard of some, like the American Cancer Society, or maybe even the American Federation for Aging Research. These big foundations can support some awards, but the majority of large awards come from the government. So through the National Institutes of Health, the Department of Defense, or other portions of governmental funding. And so in order to get any of these grants, we will write proposals. They're varying in length. They also contain all of the ethical guidelines and financial constraints that we're going to put on that project. So every single thing that we're going to buy over that period has to be line items listed. And that includes the salaries of everyone working on the project. So the university doesn't fund anyone working in my lab. I have to pay those people. So it's kind of like a small business. And I think people don't realize that my small business of research is completely funded by my work, by my own grants. The university does not provide the funds to support anyone that works in my lab. And moreover, some of my own salary has to be paid by those grants. So the university isn't even paying my whole salary. They're paying part of it. Mm-hmm. So it's a really big deal because you're kind of managing and you're dependent upon these grants to pay your students, your postdocs, your undergraduates, your research technicians to keep the project going.
Definitely. When I was doing my undergraduate research, I remember, I would hear it from my principal investigator, hey, we'll see if we get funding this semester, right? I mean, you can always stay on. Unfortunately, it would be unpaid, but if you want to continue your research, I was trying to get published with my paper and everything. So I basically had to continue, if I wanted to get that paper out, regardless if I was paid or not. I was very lucky. He was able to get funding for everyone in the lab. But it's not easy, like you mentioned. So I think, the more we talk about it, the more we understand the process, hopefully, we'll maybe see some changes or the easier it'll get as well.
Yeah, for sure. I mean, once we write these proposals, they're reviewed by scientific committees. So, our own peers are reviewing them. I serve as a peer reviewer. Other people around the country serve as peer reviewers, and they're discussed and thoroughly vetted. But if you imagine we have, a ton of PhDs, all these applicants are coming from PhDs and MDs, right? These are all very intelligent people, but only 10% of those grants are being funded. So that means that if I submit 10 grants, right, that if it were just by chance, one of them might get funded. So, it's a lot of time spent resubmitting and I'm sure you've got
and then
so
and you have to wait. Like after you submit, you wait. What is it, like 9 months, 12 months, maybe sometimes
it can
more?
be up to a year, before you actually even hear back.
That was the most surprising part that I heard. I was like, what? You mean we don't know tomorrow? We just submitted today. We don't know tomorrow, right? So they are thoroughly vetted. There's a process in place, but it definitely takes a long time, and it takes a long time to create the grants and submit them, and do all more of the kind of administration work, right? There's word limits and character counts and all of that.
Yes, and we have to be even when we get them, we have to be very careful about how we're spending the money, right? I think it's a great investment because most of the money is actually supporting researchers and jobs right here in our community. So I think that's fantastic.
Yeah. Well, it's been great, Dr. Burd. I have one curveball question always at the end of my podcast. If you could be any animal in the world, what would you be and why?
Oh my gosh, what animal would I be and why? Oh, there's so many great animals. So I am a huge fan of these cranes that come by in the water here in Ohio. They've started to appear with these really long, kind of swan-like necks. They're so beautiful. And so I would love to be this majestic crane, right? Because
there you go.
I feel like you don't have to do anything. You're just majestic, right?
Never heard it. No one said that yet. So I like it. It's unique. Well, thank you so much, Dr. Burd. We've come to the end of this amazing podcast interview. For people who, undergraduates or maybe people who want to ask you questions, where can they find you?
Yeah, so you can find me at my email address. It's Burd, BURD.25@osu.edu. And then I'll also provide some information for you to put in the links so that people can contact me if they have questions.
Awesome. Well, sounds great. Thank you, everyone, for joining the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more. Thanks again, Dr. Burd.
Thanks.